Me and my brothers have the same parents, but we're not clones of each other, nor are we clones of our parents. This is because of a process called meiosis. Meiosis is a process of cell division that produces gametes, or sex cells. The process of meiosis creates sperm and egg cells. Sex cells only have 23 chromosomes, but when they come together, they make 46, which is perfect for creating a human. Meiosis allows for genetic variety, which is why me and my brothers are different from each other. The process of meiosis is separated into two parts, meiosis 1 and meiosis 2. In meiosis 1, a special cell division reduces the cell from a diploid to a haploid. You see, a haploid describes a cell that contains half the number of a chromosome as a parent cell. A diploid describes a cell that contains pairs of homologous pairs. Meiosis 2 is similar to mitosis, but the division in meiosis 2 is a haploid instead of a diploid. Both mitosis and meiosis start with interphase. In interphase, the cell goes through the growth and synthesis phase. Chromosomes are replicated, thus beginning meiosis. A chromosome is a structure in the nucleus of a cell that contains DNA. Each chromosome is made up of a pair of identical sister chromatids held together at the centromere. The first phase in meiosis 1 is prophase 1. In prophase 1, each pair of homologous chromosomes, a chromosome that contains the same sequence of genes as another chromosome, line up side by side. This is called synapsis. In prophase 1, the process of crossing over occurs. This is an exchange of chromosomal segments between a pair of homologous chromosomes. This exchange of genetic information is the coolest part of meiosis and is an important mechanism for providing genetic diversity. As prophase 1 continues, the centrosomes move to the poles of the cell and spindle fibers form. The second stage of meiosis is metaphase 1. In metaphase 1, the homologous pairs line up along the equatorial plate, which is a fancy way of saying the middle of the cell. The spindle fibers attach to the centromere of the homologous pair. Next comes anaphase 1, where the homologous pairs separate and move to the opposite poles of the cell. This is called disjunction. The sister chromatids are still held together by the centromere. As a result, a single chromosome from each homologous pair moves to each pole of the cell. The chromosome number is reduced from a diploid to a haploid. After anaphase 1 comes telophase 1. In telophase, the homologous chromosomes begin to uncoil, and the spindle fibers disappear. A nuclear membrane forms around each group of homologous chromosomes. A cleavage furrow marks the beginning of the cell pinching its cell membrane and cytoplasm down the middle. In plant cells, a cell plate grows a new cell wall from the middle of the cell outwards to the edges, instead of pinching the cell membrane. This creates two haploid cells. After meiosis 1 comes meiosis 2. Meiosis 2 follows the same phases as mitosis. It starts with prophase 2. In prophase 2, chromosomes condense and the nuclear membrane breaks down. The centrosomes move apart and the spindle fibers form between them. In metaphase 2, the chromosomes line up individually along the equator of the cell. The centrioles help in the formation of the spindle fibers that separate the chromosomes during cell division. In anaphase 2, the sister chromatids separate and are pulled away to the opposite poles of the cell. When the homologous pairs of chromosomes fail to separate, it is called non-disjunction. This results in gametes with either too many or too few chromosomes. The consequences of this is usually quite severe and can cause conditions such as Down syndrome or autism. And finally, in telophase 2, the nuclear membrane forms around each set of chromosomes, and the cell divides, creating a tetrad, which is the forcem during meiosis made by two homologous chromosomes that have each already replicated into a pair of sister chromatids. After this, the incredible cycle of meiosis is complete. Meiosis is a process designed to create one of the most fascinating species there is, human beings.